step into the unknown and do something sometimes without knowing how the outcome will be.
There is nothing exciting about a safe bet or the middle of the road.
If you're not on the edge, you're taking up too much space.
Find the strength to overcome the challenges ahead.
There's an incredible human being left for you to discover once you reach the edge.
If you want to make a difference in life, be a scientist.
Good afternoon, everyone. I would like to give a warm welcome to everyone here who has joined us here in person and to all of those who have joined us online for today's keynote on the 9th day of WEP 2023. I hope that everyone has been attending most of the events and taking advantage of all of the different opportunities that WEP has to offer. And most importantly, I hope that you've been enjoying your time and learning lots. There are only 4 days left in this year's WEP, so be sure to take advantage of them and attend as many lectures and activities as you can. My name is Brandon Zahudnik Huntington. I'm a second-year PhD student in the Structural Biology and Engineering group of Professor Stefan Erold, and I'll be your MC for today's keynote lecture. I'm very excited to be here with you all, partly because I remember being seated where you are now, taking part in my first WEP back in 2020, but also because I have the privilege of helping to introduce Professor George Church, who is a pioneer in our field of bioengineering, among many other disciplines. I'm sure that you're all as excited as I am to hear his lecture today, discussing the diverse applications that have been enabled by the advances in DNA reading and writing. After the keynote, I want to remind and invite you all to a special screening of the film, The Boy Who Harnessed the Wind, taking place in the cinema this evening. What makes this screening special is that the boy who the story is about, Mr. William Kamkwamba, is here with us at KAUST and will be holding a book signing in the cinema at 6:15 before the screening starts at 7 o'clock. Be sure to bring along your own copy of the book or come early to try and get one there. Now, without further ado, please help me in welcoming to the stage the host of today's lecture, Professor Diana Lacoste.
Thank you, Brandon. Can you hear me? Okay. So first, I would like to welcome all of you, those in the room, but also those online. And I would like to introduce the guest lecturer, the WEP keynote of this afternoon, Professor George Church. We are all honored to have him as a keynote speaker in the WEP because he's one of the most important scientists of his generation. So Professor George Church got his PhD in 1984. He is a professor of genetics at Harvard Medical School, a founding member of the Wyss Institute, and a director of personalgenomes.org, the world's only open access information on human genomic, environmental, and trait data. Professor Church is known for pioneering the fields of personal genomics and synthetic biology. he developed the first method for the first genome sequence and the dramatic cost reduction since then, from about $3 billion to about $600, contributing to nearly all next-generation sequencing methods and companies. His team invented CRISPR for human stem cell genome editing and other synthetic biology techniques and applications, including new ways to create organs for transplantation, gene therapies for aging reversal, and gene drives to eliminate limb disease or malaria. Professor Church is director of IRRPA and the NIH Brain Project and National Institute of Health Center for Excellence in Genomic Science. He has co-authored more than 625 papers and 156 patent publications, and one book, Regenesis. His honors include Franklin Bauer Laureate for Achievement in Science, the Time 100, the election to the National Academy of Science and Engineering. So help me to welcome to KAUST Professor George Church. We are looking forward to your talk. Welcome.
Thank you very much. Hopefully you can hear me and see my slides at this point. Someone will tell me if you can't. So I want to talk about genome engineering technologies. Broadly speaking, a set of technologies we think we can apply to a variety of problems, medical, non-medical, non-biological even. And in the theme of today, EDGE, transform the world you know. So I will make many thank yous on almost every slide, and this slide is some of the institutions that have helped bring these ideas into the real world. So we're going to talk about technologies and applications. This little tRNA that's spinning here, I've been working on for almost 5 decades now, and you'll see more about that in a moment. Now, I'm going to talk about not just genome engineering, but multiplex genome engineering. There's a growing number of applications where rather than changing one gene in one organism, you might want to do many genes in a population or many genes within one cell of one organism or within one organism. So we think of these as 2 axes where you have the number of edits per genome along the x-axis and the edits per population of organisms along the y-axis. And fairly early on, we were able to get pretty high up on the edits per population of organisms. In 2009, we could make 5 billion different design genomes that were available for optimizing metabolism. And fast forward to today in the lower right, and we're getting in the changing multiple codons genome-wide, which involves tens of thousands of genome engineering changes, and we'll see that in just a second. And we'll also populate the whole plot. So in order to get tens of thousands of changes, there are a few new technologies that we think are critical. These are not only multiplex, but they're precise. A lot of emphasis recently has been on making pragmatic choices that resulted in less than precise and less than ideal edits, like the base editors and the double-strand breaks and so forth. But we're now entering or re-entering a phase where we can make large edits and precise ones. Sometimes this involves a lot of synthesis, at least for prototyping, and it definitely involves recombinases and integrases. So recombinases are a few, but are distinguished from integrases in that they involve single-strand annealing proteins, SSAPs, and single-strand binding proteins, SSBs. And this allows us to take advantage of the replication fork. And then the integrases basically are ways that viruses, phage, integrate their genome without exposing the cell to a lot of damage. Now, these SSAPs, they were limited to one organism in the whole world for engineering for decades. And we finally found the breakthrough was figuring out that there was a second protein in the cell that was exquisitely fine-tuned via the C-terminal 9 amino acids at the very tip of the single-strand binding protein. And so you'd have pairs of single-strand binding proteins, SSAPs, and that was critical. And that was just published in Nature Chemical Biology. And almost instantly, one of my companies, Inari, turned this into a technology in use in plants. It's proven in tomatoes, tobacco, and corn so far. So how can we use these tools, the SSAP tools and the recombination tools and the synthesis tools? We started in around 2013. We changed one codon out of the 64 triplet codons, 1 out of 64, and got 3-4 new properties: genetic and metabolic isolation, non-standard amino acids, a whole variety of them now, and resistance to many viruses, not all. But fast forward from 2013 to today, we think we now have something that provides resistance to all viruses, even viruses we've never seen before. This also spun off a company called Grow Biosciences, which uses non-standard amino acids for human pharmaceuticals. So anyway, as we transition from one codon out of 64 to 3 codons, it's now 18,000, and then to 7 codons, 62,000. And this is a large number of changes in a genome that's 4 million base pairs. So we have 3 forms of biocontainment. We want an industrial organism that might be used inside of other organisms in the wild or in cleanup of environment, so forth. You don't want nucleic acids to get in or out from the environment. You want it to just stay stable. And as it is now resistant to all viruses, you want to make sure that that organism itself doesn't proliferate because it has an unfair advantage over all of its cousins. And so that requires a metabolic tying it down via organic chemical BiPA that only it cannot be found in the environment. So I'll show you just one slide of data on how we confirm that this is resistant to all viruses. A previous pretty good strain from Jason Chin's lab what looked like it might be resistant to all viruses, but it turned out it was not resistant to any virus that would bring its own transfer RNA to the game. And so we specifically said to augment that by a leucine swap. This is something where we took a very potent, well-expressed serine tRNA and turned it into a leucine tRNA, or leucine encoding. So that means that every time the virus had a serine of one of these 2 codons, it would incorporate a leucine, which made every protein in the virus broken in multiple ways so that it couldn't escape, both theoretically and empirically. The host was protected, the virus died, but the host was protected because there's 6 serine codons, and we just swapped these 2 into the other 4. And this is work from Akos, Savannah, and Reagan, and others in our group. And we tested this by using all kinds of environmental samples that are rich in new viruses, including farm waste and sewage and so forth, and it passed with flying colors. Here's before and after the modification we made that shows resistance to all viruses. Now we want the host to grow quickly as well, and our previously E. coli was the fastest growing organism. Now it's Vibrio nitrogens, which we've turned into a platform that is similar to E. coli, not quite there, but it's twice as fast. And we've also established photosynthetic organisms, both eukaryotic and prokaryotic, from the wild as systems that might be able to go faster than other photosynthetic systems used for making food and chemicals. In particular, you can see that it's a 90-minute doubling time versus an estimated 17,000 minutes for some of the fastest crops like corn. So we're interested in speed of biomass doubling because of so much productivity of organic chemistry and biochemistry is dependent on that. and also food. So I'm not advocating this necessarily or predicting, but it's an interesting observation that we might be at a point where photovoltaics is about 300 times more efficient than photosynthesis, and one can convert the photovoltaics into biomass without much loss in efficiency. So something to keep one's eye on. All of it is the kind of the microbial systems I've been talking about rather than conventional crops. Now, all of this is based on these technological exponentials, where we've got factors of 2, sometimes 10-fold per year for now, a decade or more. And these are factors of 10 on the y-axis, a logarithmic plot. And for example, the cost of a human genome has decreased from $3 billion for a poor quality haploid genome to $300 for a very high quality diploid genome that is clinically relevant. And this is all due to multiplexing that we've been talking about. But what about with drugs? Okay, great, we can bring down the cost of reading and writing by 10, 20, 30 million fold, but drugs seem to be creeping up. And as an example, the most expensive category of drugs so far has been gene therapies at about over $2 million a dose. But if you take a look at the past couple of years and think about vaccines a little differently, the top 5 of all 5 of the top vaccines for coronavirus, COVID-19, are formulated as gene therapies. In fact, the cheapest one is $2 a dose rather than $2 million a dose, and it's an adenovirus carrying double-stranded gene as a vaccine. So this means that we can probably bring down the costs of many gene therapies as long as they have an economy of scale. And so we wanted to think about other gene therapies that have economy of scale. For the very rare genetic diseases, those are relatively easy to deal with genetic counseling. In some cases where you have de novo mutations, it might have to be done by in vitro fertilization, but the bulk of it can be done with genetic counseling, leaving these common diseases. So what other common diseases are there than pandemics? And most of these are so-called complex diseases or age-related diseases. And aging impacts almost every form of human morbidity and mortality. And Jalpedo de Magalis was a postdoc in my lab who established this database and has maintained it ever since. And Noah Davidson took these kinds of genes and tested them not just for biomarkers of aging, but for many diseases of aging, starting with 4 and now ever-growing list of diseases that have, the only thing they really have in common is they're age-related. And then Noah has explored 2 different collections, mixtures of therapies that handle the 10 different pathways of aging. We understand these pathways fairly well, not perfectly, but it seems evident that if you just fix one of them, you don't fix aging much. You might add a year or 2 in a public health sense on average, but you really probably have to get all of them in order to greatly rejuvenate. And so anyway, the 2 categories of 3 genes each that Noah's explored is one involves soluble factors that are exported and flow through the blood. And it's been shown that young blood can be rejuvenating. And the other category are proteins that are just in the nucleus. These are transcription factors that were made famous by Shinya Yamanaka for helping us establish pluripotent stem cells, but now they can be put in as in the form of gene therapies. Just 3 of the 4, sometimes called OSK, that have now been shown to extend life in mice when delivered at a very old, at a point where half of the mice have died already in a cohort. At that age, they can add a significant amount to their lifespan. So these are 2 different ones is located in the nucleus and one goes through blood. Now, so those are a set of powerful genome and epigenome engineering tools, multiplexed, but we need to analyze them. So I'll take a short detour into our analysis tools, which have emerged recently as extremely new capabilities. Nanopores is one, we worked on it since the 1980s. And there's two major varieties. One is the Oxford nanopore, where the DNA goes through a pore and the conductance is monitored. And the other is the Genio Roche, not yet commercially available. We've published a few papers on it, where you're monitoring incorporation of labeled triphosphates, labeled with something that affects conductance. And this has a property, many people are familiar with the Oxford nanopore, but the Genio Roche has a property of dropping the baseline for each base, rather than kind of having a complicated landscape. And so you have different conductance levels for GTA and C. Somewhat related is the idea of molecular transistors. This doesn't involve membranes or pores, but it does involve single molecule detection using sensitive electronics, where the binding of a nucleotide triphosphate, ACG and T, can be discriminated by their effect on gating the electron flow from source to drain. And again, these are capable of dropping the baseline in between events. This is from Roswell. So, and finally, there's the most commonly used sequencing, which is based on fluorescence microscopy, or so-called next generation sequencing. All of these are, but this fluorescence microscopy is the underpinning, although it's just a thin layer of randomly distributed nucleic acids. What we'd really like to do is a lot of those nucleic acids come from somewhere. They come from a three-dimensional specimen, and rather than lose that information by randomly distributing them and to retain X, Y, and Z, instead of just a monolayer 2 nanometers thick, we can have the thickness can be 40 microns or more. And so in two collaborations, one between our lab and ReadCore, now 10x Genomics, and with Ting Wu's lab, also in our department at Harvard Medical School with Bruker Vutara, these two instruments have been available for a while and are fairly mature for imaging. And they take next-gen fluorescence sequencing back to its roots of the microscopy. And now every pixel, every voxel in an image can have a sequence tag associated with it for either DNA, RNA, or protein in 3 dimensions for possibly lower cost than conventional sequencing, because you don't have to take the whole process of taking things apart and spreading them out randomly. You just take them where they are. So anyway, Rich Terry pioneered this. And I'll just show you quickly DNA, RNA, and protein. This is DNA from Ting Wu's lab at Harvard. Here are 2 pieces of chromosome 19 in human, where they're color-coded as you walk along the chain. You can walk along the folding path like you might for a RNA or DNA, RNA or protein with X-ray crystallography. Here, you can manipulate this in a similar way. There's these 2 papers from PLOS Genetics and Nature Methods from the Wu lab. And here's a kind of a zoom in of a little piece showing how high resolution this can be. This is on the order of 20 nanometers resolution, which is far better than normal microscopy. And you can see the loop closure here, where orange and red loop closure portions of the chromosome, which are 10 kilobases, and each of these 10 kilobases are broken up into 30 to 100 little individual chromatin components, getting close to a nucleosome or 2 in resolution. So that's DNA, this is RNA, this is a single cell, each dot is a single RNA molecule changing color but not position, ACGT, the 4 colors, and then the sequence tag at each pixel, each RNA molecule can be recited, and so you know what you have in each place. So that's DNA and RNA. Here's protein, where we put a 9-mer oligonucleotide tag onto your favorite antibodies. and then they can be mixed together in multiplex binding of antibodies. They can either be a full-size Y-shaped antibody, or for slightly higher resolution, you can have just a single domain, so-called nanobody. And this allows super resolution DNA exchange imaging, as I'll show in just a moment here. This is normal and super resolution of exactly the same portion of the cell. These are synapses, actually more than one cell, where, say, 4 different antibodies can agree on presynaptic to postsynaptic. You can see the little white arrows are interpretation of the directionality of the information flow, and you can tell whether they're excitatory or inhibitory. This is great for the connectome, for really understanding how the brain connects up and how it connects to other functional assays. Yu Wang did this as a shared student between my lab and Peng Yin's lab at the Wyss Institute at Harvard. So that's DNA, RNA, and protein. That was kind of an aside for how to analyze our multiplex genome engineering and multiplex epigenome engineering. So let's talk a little bit about how one can use in engineering for reading, just like we were reading DNA, RNA, protein in 3D. How about 4D, the time axis? How do we do that with in a way that retains enough information so we can do it later at super resolution, but records, doesn't lose the time axis? And this story, by the way, will involve repeat elements in just a moment. So this was, we started doing this just to encode data, meaning like zeros and ones off your disk drive, for a book that I wrote called Regenesis, here in the upper right. And so this is storing digital data in DNA, which in 2012 was a fairly new idea, and included storing the images and later whole movies, digital movies. But we think of it now as another application, rather than storing digital data that already exists in the world, is recording new digital data from biological systems that respond to developmental biology and physiological changes. And we can think of this like a digital flight recorder or DNA flight recorder, equivalent to collecting lots of data about your planes, but only bothering to read it when you're interested in a very specific thing. You don't have to read all of the flight recorders on all of the planes. And so this is similar, and now many teams have done this. I just want to call out Jay Shinduri and Seth Shipman's team. They were postdocs in my lab, and now with their own labs are really advancing this field along with other groups. I'll just give you one example of the way, give you an idea of how this can be recorded with work from Reza and Prashant. Prashant was one of the couple of teams that brought CRISPR in, as a technology, and that kind of classic CRISPR is on the left-hand side of the slide, where you make an RNA, it binds to a nuclease, guides it to another target, kills that target, does genome vandalism, makes a mess at that target, and then the target no longer is a target, and the game over. But in this new method, it is its own target. It modifies itself. The vandalism causes a new RNA to be made, and it goes through cycles of generating diversity. So we start out with about 60 of these scattered through the genome, and it just keeps changing. So this should be a worst case scenario for like causing off-target effects and unintended consequences. But these mice make it to adulthood, and they record all the way through their life what's happening both developmentally and in terms of biochemistry, physiology. And this we estimate will record about 2 terabytes of information per organism, per mouse in this case, which is only a billionth of the mass of the mouse, a very tiny fraction for all the chart recorders, for all the cells, each cell having its own. And we think we can increase that from 60 sites in the genome with repetitive elements, so we can get 20 petabytes. That's 20 times 10 to the 15th bytes of information contained in a tiny fraction of the mouse's body. Here's some primary data from another lab for the UC Irvine, which shows two interesting innovations on top of what I've already described, where they use terminal transferase to get a few more bits of information per site. And also they show that you can have a concentration and time curves encoded in DNA for small molecules or physiological events like hypoxia. And I should mention these mice are available through an NIH resource for anybody in the world at close to low cost. So, so I said that we could scale this up from 60 sites to thousands of sites. Here's 24,000 sites. Preliminary experiment how we can encode information at line elements. These are long interspersed repeats, line 1 elements. And our favorite way of modifying these are A-base editors. These are deaminases of A to G. And the C to T is more toxic, more off target. And so we prefer the A to G. And that's what we set this record in human pluripotent cells. Actually, these are PGP1 from my own body cells. And to do this, we had to be very careful about every source of single strand and double strand breaks. This is work Raphael, Chunting, Corey, Oscar, Khalid, and Verena. With very indebted to them in my lab, and also from David Liu sharing enzymes prior to publication. But we want to go even beyond line to others. We've done endogenous retroviruses in lines, but we're expanding to signs and centromeres, which have millions of copies per cell. And we've done some experiments on these now, and it's looking very promising, not only having implications for data recording, but senescence in cancer and some new applications that take advantage of the highly multiplexed editing. Now, we not only want to engineer genomes, but we want to engineer proteins, and some of those proteins can help us engineer genomes. And very often we might want to introduce these engineered proteins into genomes we want to engineer. So this is, we published about 8 papers on using machine learning plus mega libraries. Each of those MLs are separately useful, but together the ML is incredibly valuable. And the first of these was this UniRep paper, which is used worldwide now in 2019. But since then, we've spun off 5 companies that do 5 different things. Shape Therapeutics is focusing on RNA gene therapies like ADAR, an adenine deaminase. Nabla does antibodies, which we meet to custom. Manifold is developing protein tags and detectors for those, so that one can tag thousands of therapeutic proteins in a single body in a preclinical trial, so you can do effectively 1,000 times cheaper experiment if you can tag 1,000 proteins. Dino Therapeutics does capsids for delivery, and then Patch does RNA regulation at DNA or RNA level. So just give you one example from those 5 applications where we use this cycle of machine learning and mega libraries, ML, to look for immune evasion, evolve capsids that now can evade the immune system, which is good for reuse or avoiding naturally occurring immunity, and also tissue specificity. Pierce and Eric published these 2 papers in Science and Nature Biotech, and we can do over 1 million different designs at once. These are not random, these are designed by the computer. We can inject millions of different viruses into the blood and see where they home this is 6 tissues out of many that we can assess. And then you get these heat maps of viral genome on the x-axis and all the possible substitutions of different amino acids on the y-axis. And red means that it's hotter, binds better, accumulates more than wild type virus. And so you can see a lot of new specificities that you can get by this method. But that's one at a time. Big libraries, millions of libraries, but only one change at a time. What if we wanted to make lots of changes in a short region or even in a large region? We picked a region of 28 key amino acids and saw how much we could change. All of our previous work was very naive from our current viewpoint. We could barely get 4 changes and might end up with a library with 0 members in it. Fast forward to this MLML, and now we can get 60% of all the designs in a library work are viable, and sometimes often new specificities. And if you go up to 25, it's closer to 100%. Even 28 out of 28 changes, which is an amazing number of changes, we can make a sizable library. So that's how far we can go with using machine learning mega libraries to do protein and genome engineering. Now let's talk about epigenome engineering, just using transcription factors that I already introduced, the OSK that we use for aging reversal, 3 examples of transcription factors. But we have 1,700 transcription factors in our expression library now, which are available for making various combinations. So we call this collectively the TFome, and our protocols where we use combinations of these TFs, or sometimes singly. We expect and typically work until we get protocols, recipes that take only 4 days and up to 98% conversion from pluripotent stem cells to your favorite cell type, whatever it is. So we've got over 300 such tissue type recipes going from pluripotent stem cells to highly differentiated and mature. Or we can move them around to different ages, different maturity levels, pluripotent. We can make progenitor intermediates, or we can cut straight to the final target. We do this by looking at the transcriptome of the target and say, what would we have to change in terms of transcription factors to get to that transcriptome? And this is benefited by 4 companies here: Rejuvenate Bio, that I already mentioned, for aging; Gameto for making gametes; Thymune for engineering the thymus; and GC Therapeutics for a whole variety of different cell types. The GC Therapeutics came from Alex and Peristu when they were postdocs in the lab. They published this Nature Biotech paper on the 300 recipes of the TFome, including neurons, endothelium, and oligodendrocytes, which can be made into transplantable organoids that can rescue neurodegenerative demyelinating disease in mice, as a kind of a step in the direction of dealing with human demyelinating diseases like multiple sclerosis. And here you can see an electron micrograph cross-section showing this normal, the epigenetically engineered looking normal myelinating, just like happens in your white matter in your brain and the spinal column. So this is showing that it works at ultrastructural level, and this is showing it works at a behavioral motor function test in the lab. We've not only looked at the oligodendrocytes, which the immune system damages, but also the immune system itself, including cytotoxic and regulatory T cells, microglia, immune cells in the brain, and a variety of other cell types, thanks to Evan and Sung Lei. And Sergey has developed, recently entered the lab with technology to do super socks, again, these transcription factors that are important for reversing of aging and establishing pluripotent stem cells. Another application of the TFome is in getting gametes and in cells that support gametogenesis. For example, the granulosa cells, which support the primordial germ cell oocytes precursors that Christian Cram and Merrick Smela have done. And this spun off Gameto for helping in vitro fertilization using granulosa cells to help mature immature eggs. And and Orchid Health is providing another assistance in vitro fertilization, where every single IVF embryo can be sequenced, whole genome sequence, very high quality. Something was unthinkable, a decade ago. Now is routine in IVF. Another example of multiplex editing that is making its way into clinical trials is engineering animals so that they can provide organ donors. This requires, we collected all the wish lists from all the people in the field that have been accumulating since 1963, but didn't have good editing tools. Now they have good editing tools. We did all 42 at once, including changing the sugars on the surface of the cells and various blood functions, immune functions, and importantly, getting rid of all the endogenous retroviruses, which are very active in pigs and produce viruses that will infect human cells, which is particularly bad in immune-compromised patients. So we got rid of those, and those protocols are well known, published, and now we have preclinical trials getting up to 600-day survival of various transplants. We were testing kidney, liver, and heart initially. Jim Markman is one of several hospitals, leading one of the teams at Massachusetts General Hospital. And Luhan Yang was a student and postdoc in my lab who co-founded Keyhan and eGenesis to bring this. And then the very last example is what we can do for the environment, not just for medical and veterinary applications and agricultural applications, but in the environment, we want to help endangered species, restore them to very healthy populations, and we'd like to also help ecosystems. Sometimes a single keystone species can have a big impact. For example, kind of classic now example, the wolves in Yellowstone, they were missing for 70 years and were reintroduced with expectation that they would have a big positive influence. They by attacking a certain large herbivores, not reducing their population, actually ultimately increasing the herbivore populations, but driving them away from the young trees so they could grow into big trees, including this willow population that was measured in Owen Muller's paper. And once those were restored after introduction of the wolves in the 90s, the willows restoration resulted in beaver being able to do their job, which resulted in changes in rivers and lakes, and amphibia, fish, and aquatic birds. So this wolves had big ramifications, positive ones for the whole Yellowstone Park. A similar thing I think exists in the Arctic, where the relatively recent, 10,000 year ago, elimination of major herbivores in the Arctic has resulted in a degradation of the environment, both in terms of diversity and in terms of human needs of keeping the Arctic carbon frozen. So there's 1,400 gigatons of carbon at risk out of compared to only 10 gigatons, which is a human annual consumption. So if this escapes, and you can see here our friend and colleague Sergei Zimov demonstrating just how much methane there is coming out of the soil and in the lakes. Methane is 80 times worse than carbon dioxide, so you really want to keep that frozen. And there are three different ways that the ancient herbivores kept the soil cold, which is reflection, better photosynthesis, and allowing access to snowpack. The elephants, or mammoths in the old days, are one of the few herbivores that can knock down trees easily, and they love doing it. We pursued this at a very low subsistence level for a decade until Colossal came along, and now we have adequate funding to engineer elephants to reduce their exposure to killer viruses, which is making them endangered, as well as providing them with new environments where they can have an impact. So my final thank you for this particular last slide is all these teams that have kept the flame alive until Colossal came along, and now it's growing by leaps and bounds. So that's this little quadrant of de-endangerment and xenotransplantation. Period, full stop. I'm open for questions if there are any. Thank you.
Thank you very much, Professor Charles. It was really an inspiring talk. We have a time for a shorter Q&A session. If you have a question in the audience, please go to the microphone and yeah, you can start. Introduce yourself first, please.
Hello, everybody. My name is Alexander, and I'm doing PhD in computer science, but I found the talk amazing and very interesting. I would like to ask, we saw that there was a huge progress in developing these techniques, also from a price perspective, as well as from editing capability perspective. So my understanding is that we will first apply these techniques for humans, for diseases, and to remove all the imperfections. But my question is, are we going to be targeting enhancing the human capabilities? For example, if somebody has improved memory capabilities, and we can isolate the changes, let's say using some of the techniques with artificial intelligence to see which differences between people with better memory versus with worse memory. And do you see this coming to a play in the future? And the second part question would be, what impact would this have to the human society?
Yeah, that's a great question. You know, a fair amount of my publications have been on ethics and safety. And this topic comes up. I think that we will, that most of our technologies these days actually are enhancements relative to our ancestors, whether they are biologically based, like vaccines, are a huge enhancement relative to our ancestors lived in fear of you know, dozens of different killer pathogens. So we would appear to be superhuman compared to them. And the same thing goes, when we discover something in the population that might give an advantage, we use that as a starting point for example, developing gene therapies that could help an adult be resistant to viruses, resistant to cancer, to senescence, and so forth. And I described how we're doing that in dogs and human, using these cocktails of 3 now, probably more genes to get you know, longer, youthful physiology. So enhancement by itself, I don't think is the alarming. I think what people get concerned about is enhancing the germline. And this is, I think, no strong argument is made in favor of it, and many arguments have been made against it. Basically, essentially everything that we could do in the germline, we can do in after birth. You know, and many of them in adults, where we now know what the individual needs are and capabilities are, and we can make a more intelligent decision as to how to enhance them. So I think enhancement is fine in most cases, as long as it gets the scrutiny that we apply to all drugs via the FDA, the EMA in Europe, and other similar organizations throughout the world. If they can all agree that it's safe and effective, and that's a big step. And then the other ethical component is making it affordable, which I've highlighted in my talk, is I think we can make these affordable as well. Thank you for that question.
Thank you for the answer.
Thank you.
Thank you. Good morning to you. Maybe my question is similar. We have noticed during the COVID-19 pandemic that the vaccinations have been approved by association like FDA and others in no time. While we know that during the previous years there is restriction on testing on human, whether it's ethical restrictions or maybe the risk or the side effects of any of changing the gene or engineering it. So my question is how do you think these restrictions holding the progress and improvement in this field to reach its edge? Thank you.
So if we're talking about the rate at which the FDA and similar organizations around the world approve drugs, approve drug trials, occasionally it seems slow, and when it really mattered, it was quite fast. When we had a pandemic, we were getting drugs approved, vaccines approved in 11 months for emergency use. So it can be done quickly. There is some risk there, but the feeling was that accelerating it from 10 years to 11 months, the benefits would outweigh the risks. Now, these were new categories of vaccines, so it really was not just changing the seasonal flu, just some subtle amount once a year, but it was a big change, and it worked. And I think it changes our attitude towards gene therapies in general, since these were formulated as gene therapies. It doesn't mean we should just approve all gene therapies in 11 months, but it does show that we can do it. But I think we need to be supportive of these agencies and be supportive of them being careful, even if it does take time. Certainly in between pandemics, in between crises, we should use that time to get ready for preventative medicine. Thank you.
Hello, my name is Fabio. Thank you very much for this talk. And my question was about the data storage inside the genome that you talked about. I think you talked to up to 2 terabytes of data storage. So what are some other uses of this data storage, and how do you think it might be applied to the world as we know it?
Yeah, good question. Some non-obvious applications that I didn't mention. Once you can do physiology, you can do physiology on, say, the eye, where each cell in the eye is a flight recorder, a DNA recording of the photon flux as a function of time, because photon affects physiology, which can then be turned into a DNA change. If you can then store that grid, it's just essentially it's XY plane with a time axis in DNA, then all sorts of small and large organisms can record what they're seeing in DNA without interfering with the vision. And then once they die, then you can harvest their retina and figure out what was happening in X, Y, and time. So in a certain sense, you can think of these as mobile, autonomous video recording drones, much quieter than typical drones, much smaller, and potentially ubiquitous. So I think this is something that we need to talk about in advance to decide, do we want this, under what circumstances, how high a priority is it, and so forth. But I mentioned that as one of many things that one can do biologically that are challenging to do with current electronic circuitry.
Thank you very much.
Dr. Church, thank you for the talk. I have a question about CRISPR. I heard about it, and I see that there is quite a bit of excitement about its potential. And can you explain what it is and why should we be excited to a non-specialist?
Yeah, I may not have emphasized it as much as I could have because there are alternatives that precede it and alternatives since then. some of which don't have anything to do with CRISPR. And I mentioned a few of those, partly because I knew you were all going to know something about CRISPR. I think CRISPR, a way of thinking about it is that it is iconic. It's like, the Beatles were not the only rock and roll star in history. They were iconic. They were an example that captured the time. And I think the same thing is true for CRISPR. Is there are many ways to do genome engineering. That's why I refer to it by the generic term, genome engineering. And another key component of this is being able to read genomes, because you really can't write genomes or edit them if you can't read them. You need to read them in advance to know what you want to change, and then read them afterwards to make sure you got them and not too much off target. And to read the epigenetic components as well. So I think CRISPR is part of a bigger revolution. That's the CRISPR technology hit in 2013. We published a paper, and then 3 months later, there were 10,000 labs that had requested the plasmids. But it's embedded in this bigger context of reading and writing genomes, which is exponential. Thanks.
Thank you.
Maybe there is no more question in the audience. Can I ask you, what was the turning point in your career? Something that you now think that it changed everything? It was someone you meet, some result in the lab. What was this point for you?
Well, there have been a number of such dramatic events. Some of them are like, I'll pick one that could be construed as negative, that turned into a positive, kind of like the phoenix rising up out of the ashes. I was heavily involved in structural biology as an undergraduate and starting graduate school, but I was so excited about the experiments that I flunked out of graduate school, but fortunately I was picked up a year later at Harvard, and I restarted my PhD. And started with Wally Gilbert, who 3 years later would get Nobel Prize, and so I was there for all of that. And his lab was basically about reading and writing DNA, not quite genomes yet when I started there. So they were part of the recombinant DNA revolution and part of the DNA sequencing. So that was probably a turning point that required me to leave structural biology temporarily anyway, and move to molecular and cell biology and computer science.
Okay, thank you very much. Thanks. So we have to stop now. Thank you so much. It was really interesting, really inspiring, and hope to meet you in person one time at KAUST. Thank you all for attending this session, and see you soon. Bye-bye.
Take care, everyone.